All right, welcome everyone. This is another of our Astina Talks. Astina Talks is part of a regular meeting event series where we try to present researchers and their latest research, including award-winning research like today. A little bit more about Astina before we get started. Astina is a North American research organization trying to bridge transatlantic collaboration between Austrian researchers and North American researchers with a focus on helping us establish networks on both sides of the Atlantic. And part of our mission is to trying to highlight the amazing research that is carried out across the many different fields that our members represent. And so it's a pleasure for myself to introduce today's speaker, Alexander Lurcher. I'm not a biologist or medical professional, so I'm going to try to do my best at introducing Alex. But if I mess up, please, Alex, let me know and feel
Sure.
free to correct me. So Alex is an immunologist interested in how infections leave lasting imprints on the immune system and shape our responses to future diseases. He completed his PhD in Vienna, where he studied how viral infections reprogram metabolism and immune responses. And he's now a postdoctoral fellow at Rockefeller University in the lab of Charles Rice, and soon to be at Heidelberg University, if I understand correctly.
Yeah.
His current work focuses on how the innate immune system can develop a kind of memory of past inflammations, even without recognizing the same pathogen again, and how this memory influences outcomes of later infections. As part of his research line, I want to highlight that Alex was honored with the 2025 Astina Award for Outstanding Scientific Publication. There's a recognition given by Astina, given to Austrian researchers and Austrian affiliated researchers in North America for exceptional research contributions. And so I'm very excited to have Alex present his award-winning work today. So he'll share some more insights on his work, and I'm very excited to welcome Alex. Alex, please take the stage.
Yeah. Thank you very much, Lucas, for the kind introduction, and thanks for Astina having me, and for all of you for being here. Good afternoon or evening, wherever you are. As Lucas mentioned, I'm gonna talk today about like a topic that I've kind of spent the last 4 to 5 years of my postdoc on, and which is revolving around how pathogen-independent immune memory can protect us from future diseases. So I want to start out very broadly and kind of give a brief intro of how we classically viewed the immune system and kind of divided it into an innate arm of immune responses that consists of cells such as, for example, macrophages and neutrophils. And this innate immune response is very rapid because it's based on pattern recognition. So those cells, they recognize pathogens not by very specific entities, but kind of broadly recognize viruses because they all share a certain molecule, for example. So this means this response is non-specific, and people for the longest time have thought that this does also not result in memory formation or immunological memory formation, because that was a trait that was always attributed to cells of the adaptive immune system, which are T and B cells. Those T and B cells, they take approximately 7 days to get fully activated. So it's significantly longer compared to like hours and days compared to the adaptive innate immune system. And part of the reason for that is because they do recognize very specific molecular entities of pathogens. So they really specifically recognize, for example, influenza A or SARS-2 and not any other pathogen. So it takes a while to kind of get this response going. But the benefit of this is that it's, on the one hand, highly specific and robust, but it also allows this formation of long-lasting memory, either mediated through T cells or B cells through antibodies. However, over the last probably 10 to 15 years, people have kind of described and done quite some research on this phenomenon of innate immune memory, which is a phenomenon where if innate cells or innate immune cells such as macrophages encounter a primary stimulus, such as a virus, they would respond to a certain degree, as indicated here in this cartoon. After the pathogen or threat is cleared, they enter like a stage of homeostasis or baseline again, where they do not actively respond. But then if they encounter another stimuli or inflammatory event, in the secondary activation, the response will be different. Here I depicted as an increased secondary response, but this doesn't have to be the case all the time. And the difference between this primary and secondary response is what we would refer to as innate immune memory. And really the basis of this innate memory, which is now, because it's not pathogen specific, it's kind of broadly effective. The basis of this is epigenetic remodeling that is retained in this basically seemingly resting phase of cells. After this first activation, they kind of retain epigenetic remodeling that then allows them to respond differently the second time around. And what do I mean with epigenetic remodeling? To kind of just a very brief detour of how DNA and chromatin is organized within a cell. I think we all know that the DNA is located in the nucleus of a cell, where it's wrapped around proteins and then eventually forms chromosomes. Have a very characteristic shape. But if you zoom into a chromosome, you can see basically that the DNA is wrapped around proteins and can be more tightly packed, like here in the case of heterochromatin, more loosely packed in euchromatin. So here you can see where the DNA kind of depicted as a string, basically wrapped around these spheres of protein, is more loosely packed. But then kind of when DNA is completely unpacked, so without those proteins, it's really accessible for DNA binding factors. And those DNA binding factors, such as transcription factors, they then allow active gene activation and transcription. So this means these open regions, they would represent active genes that are fully accessible to be activated and transcribed. Regions that are tightly packed, such as here, for example, they are generally inactive genes, so the cells do not actively express them. And then there are certain regions that are more loosely packed. So there's still some kind of proteins associated with the DNA, but they are more readily accessible. And those would be loci, for example, associated with this innate immune memory, where they're kind of ready to go and to be expressed again in the second wave of response. And what we wanted to know in this project was whether in the context of commonly circulating respiratory viruses, whether past respiratory infections actually impact subsequent heterologous infections with a completely different virus. And then kind of the long-term motivation was whether we can understand this phenomenon of innate immune memory well enough to eventually aid development of broadly effective new therapeutics. And the model that we chose was a model of SARS-CoV-2 recovery in a mouse model. So we had a mouse-adapted strain of SARS-CoV-2. We challenged normal laboratory animals and let them sit and recover for 30 days. So in red, you can see mice that were infected. So they do drop quite some body weight upon infection, but by 30 days, they kind of again catch up to the initial body weight. And wild-type mice, so uninfected mice in black, they kind of stay the same over time. We confirmed that those mice after 30 days, they are negative of virus, so they fully cleared the virus. So this is just a picture of a lung histology slide. We can see brown speckles here on 3 days post-infection. So acute infection, you can see these brown speckles that are SARS-CoV-2 antigen. But in naive and recovered, you basically don't see them. So the mice are fully cleared. They fully cleared the virus. They also, like we confirm with other parameters such as active immune response, etc., they are behaving like wild type, basically, or uninfected animals. But now we wanted to know whether, like what I just told you, whether actually cells of the innate immune system within the lung following this initial infection do retain this chromatin remodeling or epigenetic reprogramming, which could lead to the formation or could lead to innate immune memory. So what we did is we flushed out the immune cells of the lungs of either naive in black or recovered animals in red. And then we performed an assay that is called ATAC-seq, where you just can assess which regions of the genome or chromatin are more open. So you basically read out open DNA stretches. So you know which genes are in theory accessible or not. And we did this on a single cell level. So basically we did this for every single cell within the lung that we isolated. And we identified a lot of different cell types within the lungs of naive and recovered. Not too surprising, but half of the cells were macrophages. So these are these innate immune cells that have previously been shown to be able in principle to mount this innate memory. And already here in this clustering, so this is kind of unbiased arrangement of cells, just how the group cells together that have a similar epigenetic profile, you can see that the macrophages do come in two flavors, and they are just agnostically named them cluster 1 and cluster 2 here. So this already implies that there might be something happening in terms of epigenetic remodeling. So indeed, if we, as a first check, if we just color code those dots, so every dot is a single cell by sample origin, in black would be naive, and in red recovered, you can immediately by eye see, I think that cluster 1 is kind of a mixed bag of black and red cells, so naive and recovered, whereas cluster 2 is almost exclusively composed of recovered cells. And this already tells you, like on this very unbiased analysis, that there is some durable epigenetic remodeling retained 30 days after SARS-2 infection. So now we can go one step further and then ask, okay, which regions are specifically associated with recovered versus naive macrophages? And in red on the right, I'll show you what is enriched in recovered, and on the left, what's enriched in naive. And this is basically an analysis where we ask which DNA binding factors or transcription factors are likely to bind to these open regions. And we can see that in recovered, we found a lot of IRF and STAT proteins. So these are DNA binding proteins that are all associated with antiviral innate immune signaling. So every cell in the body would kind of switch on those proteins to produce antiviral genes. And to kind of in a nutshell describe this to you, if this is a cartoon of a cell, basically, if a cell encounters a viral RNA or poly-IC, which I will use later. So this is just a molecule we use in the lab to mimic a virus. It's not an actual virus, but the cells respond to it as if it was a virus. So a cell would recognize those foreign molecules or patterns of pathogens via RIG-I-like receptors, for example, in blue. And this will then activate one of those IRFs, and in that case, IRF3. So this IRF3 transcription factor will then go to the nucleus, bind DNA, and initiate the expression and production of type 1 interferons here in red. And those type 1 interferons, after they are secreted, they do bind to their receptor, which is called IFNAR1. And downstream of this receptor, again, transcription factors or complexes consisting of STAT1, STAT2, and IRF9 get activated. They go to the nucleus, and then they induce a whole range of antiviral genes. So this is hundreds of genes that are known to have antiviral properties. Genes belonging to this family are, for example, IFID1, NX1, or ISG15. So this is kind of antiviral signaling in a nutshell. And what we now wanted to know next is, because we kind of, our analysis on a single cell level, predicted increased chromatin or DNA accessibility of those transcription factors like this IRF3 and STAT, whether the downstream genes, such as these interferon genes, IFID1, MX1, are they actually also transcriptionally poised? Do they retain some memory marks following SARS-2? And for that, going back one step, we used a new method, or kind of a phenomenon that has been described like a couple of years ago, where not only is it known that in steady state, chromatin of response genes is highly condensed, so they're inactive, and in acute stimulation, they open up and get more loose. But there is also a so-called histone code to that. So I told you before that the DNA is wrapped around these proteins that are called histones. And those histones can be modified by chemical entities such as methyl groups or acetyl groups. And those methyl groups, or acetyl groups, depending where they are on the histones, we can kind of assess or investigate whether this is an actively transcribed gene, so active gene, or a non-transcribed gene, a switched off gene, or a memory gene. And for memory genes, people have previously described, like maybe 7 years ago or so, that there is a quite specific chromatin mark for those memory genes that are not actively produced, but they are kind of ready to go. And this mark is called H3 K4 monomethylation. So it's not really important what the name is, but it's like, this is one mark that we now can use to really assess whether these antiviral response genes are really kind of poised to be transcribed in a second time. Are they kind of memorized from the first infection? And what we did is to assay that, we isolated macrophages from recovered and naive mice as a comparison. And then we performed, on the one hand, bulk ataxic. So this, again, this assay to just read out open chromatin regions. But then on top, we also performed a method that is called cut and run to really look at this specific memory mark. And I'm showing here just one gene as an example, which is an interferon stimulated gene, so a known, well-characterized antiviral gene that is called MX1. this gene is transcribed or produced from the right to the left. So here would be the transcription start site in this region. And you can see on the attack-seq track on top that recovered cells here in red, they do have more signals. So this means there's more accessibility of this transcription start site, which makes sense. Is in line with our other data. But then this increasingly accessible region is also increasingly decorated with this memory mark of H3K4 monomethylation. So not only is it more accessible, but it also those histones really also bear this mark of kind of this bookmark of antiviral gene response, which would in theory allow them to respond better the second time around. Just as a comparison, if we look at a general inflammatory gene or NF-kappa B target gene, such as CXCL1, you can see there's basically no difference between recovered and naive, and if at all, there's even a reduction in recovered. So it seems that there's like a kind of a bias towards an antiviral memory in this case in those cells. So now obviously that we kind of went in depth characterizing this epigenetic imprinting, we also want to know next, so does it actually lead to any consequence? So is there a functional memory or not? Or is this just something that happens but has no impact at all? And to do that, we again isolated macrophages from naive and recovered animals. And then we in vitro, so we take the cells out into cell culture out of the mouse and stimulate them with different PAMs mimicking different pathogens. And we chose PAMs to mimic a virus or bacteria. And then we assayed basically gene induction of response genes. And what I'm showing here in this heat map looks complicated first, but it's actually simple, I think, is that we have three groups. So we have control stimulated cells. So this is basically baseline. Then we have poly-IC re-stimulated. This mimics a virus. And then we have LPS re-stimulated cells that mimics a bacteria. And then we always have 3 samples of naive and 3 samples of recovered for each condition. Each row represents expression of 1 gene, and the top 6 genes are antiviral genes, and the bottom 6 genes are NF-kappa B or generally inflammatory genes. And the more yellow, the more induced the gene is. And so I think just by eye, it's very obvious that that those recovered cells are really hyper-responsive in terms of induction of interference-stimulated genes or antiviral genes. So they are just much better. If you re-stimulate them, they induce those genes to much higher levels than their naive counterparts. And interestingly, this was not the case with general inflammatory genes. So if you stimulate those cells with LPS, both naive and recovered induce those genes, but there's really no difference between naive and recovered. So it didn't matter if the mice were previously infected with SARS-2. So there was no memory there. So what we found with this experiment was that, yes, indeed, there is epigenetic, there is innate immune memory being formed after SARS-2 in those cells. And this seems to be specific for antiviral responses. Indeed, if we do a more functional assay, so not just gene expression, if we take out those cells and then again in vitro, so out of the mouse, we infect them with a reporter virus. So this is a virus that basically lights up green. So you can just look at the cells and read out how many green cells do we get if we infect those cells. We can see that in uninfected, there is 0 infected. That makes sense. But then if we infect the cells in black, you can see that naive cells, they are way more susceptible. So approximately 40% are infected with this virus, whereas for recovered cells, we get only 20%. So it's a reduction by 50%. So also functionally, this memory has an impact. But does this also, like in kind of in an in vivo context, like in an organism, does this also impact disease of a secondary infection? And for that, we again went back to our mouse model. But what we did now is that after recovery from SARS-2, so we let them sit for 30 days again, we now challenge those mice with influenza A virus, and the strain is called PR8. And this is a sublethal dose, so the mice will lose body weight. And you can see here that in blue, mice that were previously naive and were infected with PR8 or influenza, they do drop quite some body weight by day 9, whereas mice in red that had previously recovered from SARS-CoV-2, they almost lost no body weight. So they were really protected against this influenza A-associated body weight loss, which is a disease relevant readout in this model system. So that's cool, but does this really is this really driven by memory in macrophages? And for this, we did a depletion experiment where, again, after recovery from SARS-2, we then used a chemical compound, which we administered intranasally to really only deplete macrophages within the lung of mice. and then we challenged mice with P8. So now we have a group where basically we kill off those macrophages and look if the memory is still there. And what we found is that this looks complicated, but this is again body weight loss, and I'll walk you through it. If we look at the control groups here in blue, full squares, so this is the control group, naive and influenza A infected, so they lose body weight. In orange, full circles, that's our SARS-2 period, so they almost lose no body weight, just as I've shown you before. But now if we treat both groups with this chemical compound to deplete macrophages, this difference basically collapses. So what this tells you is that now both groups are equally susceptible to influenza A virus, telling you that you really need those macrophages for this memory effect to occur. But are those macrophages also necessary? So for that, we did a bit of a more complicated, technically more challenging experiment where we isolate naive and recovered macrophages from mice, and then we transfer them into a new mouse. So this new mouse has never seen a pathogen. It just received either cells from naive or cells from recovered animals. And now we challenge this mouse with influenza A virus and look at body weight again. And again, we find that mice that had received recovered macrophages, they did better in terms of body weight loss compared to mice that received naive cells. So this tells you that this memory in those macrophages is also really sufficient to at least to some degree, reduce disease severity of influenza A virus. How potent is this? Can we also prevent very severe cases of influenza A virus in mice? Yes, we can. So if we challenge mice that were recovered from SARS-2 with a lethal dose of PR8, you can see that in blue, by day 10, all mice that were previously naive succumbed to disease, whereas all of the mice that had previously recovered from SARS-2, they are all still alive at this point in time. So this just highlights how potent this phenomenon is in terms of preventing severe disease. And it's also very well correlated with, here in blue, naive mice. If they infected with influenza A virus, they show like a very steep increase of neutrophil influx into the airway, versus mice that had recovered from SARS-2, much less so. And this is also reflected by increased cytokines or pro-inflammatory cytokine presence in the airway of naive and infected animals, which is really a hallmark of influenza A virus disease severity. Because in this model, much like in humans, mice succumb to disease not because the virus kills cells, but because the immune system doesn't manage to control the virus initially, and then it just goes wild, and the mice actually die due to this excessive inflammatory response. So what I hope I could show you now was that we found that after primary infection with SARS-CoV-2, following recovery, that alveolar macrophages within the airway, they do mount antigen-independent immune memory or innate immune memory, which allows cross-pathogen protection, which in the case or context of a secondary influenza A virus infection reduces disease severity and cytokine storm by an increased antiviral activity and interferon response. So now I decide to also, because usually people, when they see this, they say, that's nice, but what about humans? So we also, I decided to include this data here because I thought it's kind of nice, because we managed to get our hands on a very interesting cohort of patients where we could assay whether in patients we also see a similar epigenetic program. And this cohort of patients, while I acknowledge that there is like a big difference, like the comparison is difficult because we have a very reduced number of patients. If we look at slightly different cells, but I think still there's some value to this kind of analysis, because the patient cohort that we got was like from April 2020. So this was after the first wave of SARS-2 in New York City. So this means that the healthy controls are really healthy, non-vaccinated, no past SARS-2 history. And the recovered patients, in that case, they recovered from a mild case of COVID-19. This was their first infection, which is two groups that nowadays you cannot get anymore. So I think that's why I think this analysis is particularly interesting. And what we did is we looked at peripheral blood cells. So we couldn't look at lung cells for obvious reasons. But if we look at peripheral blood cells and perform again this attack-seek approach where we look at open chromatin or accessible DNA structures, if we focus on myeloid cells that are resembling macrophages in mice, we find that the subset of CD14 positive cells here in red they really closely, or most closely resemble the gene expression signature of our macrophages in mice. Again, if we look at the chromatin accessibility in those cells, we can see that in recovered patients, we do get increased accessibility of transcription factors belonging to the IRF family, again suggesting increased antiviral response or memory. For my last slide, I decided to kind of zoom out again and kind of make a point why we actually, or why I care about this and why we should care about this. And that is because immunological memory is undoubtedly the basis of herd immunity, or maybe herd resistance is even a better term. If you imagine like same scenario that happened like 5 to 6 years ago, if a population encounters a new pathogen with the right properties in a naive population, a single infected individual here depicted in red can spread this pathogen to every other member of this population that is susceptible. And this is here depicted in blue if it's a new pathogen, for example. So this will lead to a severe health and socioeconomic burden on the whole population level and might warrant some like very drastic intervention measures such as lockdowns, etc. Whereas if you have a scenario where a sufficient amount of individuals within the population are either resistant to infection or are less good at spreading the disease or developing severe disease, this spread will be significantly slowed down and you will like have no need for like drastic intervention measures. And I think the whole motivation for this is, or what we kind of want to get at in the next years, I think, is whether we can harness this phenomenon of innate immune memory and understand it well enough to either bolster existing therapeutics to be more broadly effective, or to even develop new strategies to achieve herd immunity against emerging viruses, which we don't even yet know about, because this phenomenon would allow you to basically design a therapeutic which is broadly effective. So it doesn't really matter what the next pathogen will be, because we know it will still be effective to a certain degree, at least. And with that, I'd like to end. I'd like to thank the funding agencies, Rockefeller, NIH, and the HFSP. I'd like to thank Charlie, my postdoc mentor, collaborators in New York and beyond, the Josefowitz Lab, Artis Lab, Rosenberg Lab, Rohan Parsa, and Luis Chiriboga. And now I'm happy to take any questions, and thank you for coming today and listening to me.
Thank you so much, Alex. That was a fantastic talk. Super,
Thank you.
super interesting, very relevant right now. Yeah, very clear language. I got 20% of it, so I'm happy with that. So, and I think that's awesome. And I already see some people have their hands raised. So we can proceed right to the questions. Let me see. I know there is, I think I can see the order somewhere. I think, Lucas, you were the first one to raise your hand. So please go ahead.
Hey, thanks for the nice talk. Enjoyed it very much. I have two questions. So one was for the very first part where you showed these two macrophage 1, macrophage 2 populations in your ATAC UMAP. I was wondering, have you also looked at the proportions of tissue resident macrophages in those two versus the monocyte-derived ones that would influx during the inflammation?
Yes, we looked at this quite in depth. I didn't put it now, but so what we believe that, I mean, there is some, if you get severe lung disease, there is like a certain degree of infiltrating monocytes. It's kind of an emergency response, right? We did do quite some data, like bone marrow chimeras labeling experiments that all suggested there is some contribution, but it's really limited. So it's like maybe 10% of those cells are like infiltrating cells. And I think like we did like the genetic, like the transcriptional profiling. So we do believe there's very little contribution actually of monocyte infiltrating macrophages in our model. But we did not further, like we did not like sort them out and then profile them separately. So this is like all like correlative evidence, but I think we did like 2-3 lines of evidence that suggest that at least to some degree, or to the majority, I think is like really intrinsic tissue resident memory. And there is probably some contribution of infiltrating monocytes, but I think it's not the major driver.
Cool. And maybe one little question. Have you looked at the location of those by staining for this H3K4ME1 to see whether there are distinct tissue niches, that might keep their pro-inflammatory status up?
Yeah. No, we didn't do that, but I think that would be very exciting, like to get like a spatial resolution of like basically what is the environment that maintains memory? Kind of this gets at this question. But I think that would be very, very exciting because I could imagine that like it's like not all cells behave the same, but like there's certain niches that favor those memory cells. Or it could even be that, for example, those memory cells, they're longer lived now, and that's why they kind of carry over the memory. I think we don't know enough yet to kind of what maintains the memory, but it would be very interesting. Yeah.
Yeah. Well, there seems to be a good antibody out there, so can be done. Nice.
Yeah.
Thank you.
Thank you.
Thomas, do you want to go next?
Sure. Thank you very much. That was great, Alex.
Thank you.
I was just wondering, there's like also two questions I have. The first one is, what actually causes the memory? Is it just like PAM stimulation, or is it cytokine signaling or anything? And the other thing is, how sustainable is the phenotype? Can you like just culture them in subculture them a few generations and still have the same effects?
So we think there is at least two signals that you need. So I think you need PAM recognition and type 1 interferon signaling. There have also been other studies that suggest that. So that one or the other is not enough. I'm also not sure. I think that's still open. If in vivo, you also need a certain degree of tissue damage. Because, for example, if you just treat mice with poly-IC, you don't really get the same memory like you get here. So it's much weaker if you find it at all. So there's maybe another component.
I'm sorry.
I didn't try heat inactivated yet. No. So I think there might be another factor as well, like tissue damage or a damp that is released upon cell damage. The other thing is, yes, I mean, there is models for alveolar macrophage culture. So we didn't do that. Like we could take out the cells and culture them ex vivo for some time. One limitation for this study was because back then SARS-2 in the US was still considered BSL-3. So this would be a more challenging experience, but now it would be easier. But what we did, however, is like to just model this in vitro using wild type macrophages. So if we expose them to different PAMs and then let them rest and then try to infect them or re-stimulate them, we do see a similar memory effect. So you can study this in vitro as well. But again, this maybe goes back also to Luca's point before, like this is a model. So I think like in vivo, it might be quite different because like also I just told you that in vivo, I think poly-IC alone is not, at least in my hand, was not sufficient to induce memory. Whereas if you just do it in vitro, it seems to be sufficient. So there might be some quirks to like where the cells are localized or to the environment as well.
Yeah, it might also get like dampened over time, right? Like after.
Yeah, I mean that's for sure. Like if we let those mice sit, so I only showed experience with 30 days recovery. So we still see the memory if we let them recover for 60 days. But then we dragged it out for 100 days. It's almost gone, and then I think the next was 200 days, and then it was definitely gone. Okay. So I think it's most effective in the range of like 1 to 2 months. But again, I also want to point out here that this is laboratory animals, so that they're in a very controlled environment. So once they recovered, hopefully they don't see any other pathogen because it's like a pathogen controlled environment. Whereas in humans, it would be very different because we are like exposed on a daily basis to like thousands of pathogens. So we would never enter this stage of like no exposure again. So I think in humans, maybe it's different because you kind of always re-trigger your trained macrophages or not. So I think that's one thing we also don't really know yet, I think.
So would you suggest to get a live vaccine every year just to trigger your innate immune memory to whatever pathogen you might get?
Well, I don't think it needs to be necessarily live. There was actually a paper that came out maybe yesterday or 2 days ago that showed it doesn't need to be a live vaccine, but it could also just be a combination of, a strong antigen with the right adjuvants that can like convey broadly protective effects. So, I mean, I don't want to make a statement everyone should get a live vaccine now every year, but I also think it doesn't hurt, for example, to get the flu shot every year, because I think in the worst case, it will just overall boost your ability to fight pathogens.
Yeah. Thanks. Very interesting.
Thank you.
Christine, do you want to go next?
Hi. Yeah, thank you so much for your talk. That is insanely interesting because I work with SARS-CoV-2 in the mucosal immune system context.
Nice.
I have so many questions. You answered a lot of them during your talk. So two main ones stayed, but probably going to shoot you an email at some point.
Sure.
First, did you check the complement system? if any factors there are maybe upregulated that also stay there for a while? Because
We
that's
didn't do
also
that. We didn't do that at all. No.
okay. Because
We know that, like, I know from the RNA-seq that the macrophage, like the recovered macrophages, they do upregulate like factors of the complement system, but we've never measured the protein or did like any functional assays. No, we did not.
okay. That might be interesting though.
I mean, I'll look at it. Yeah.
Nice. I got another question. Because you said you were looking at sections of the chromatin that were more accessible, and I wanted to ask, were you looking into DNA methyltransferases and how they were upregulated or downregulated? Because I think you also did single cell RNA sequencing.
No, we also didn't do that. That's also something that's still on the list. I think we very much focused on chromatin and histone code, but not so much on the DNA methylation part. But I do agree that this could be also like the next regulatory layer, which we don't know about yet. I think there's actually very few studies that did this in context of brain immunity, like looking at DNA methylation. But I do think that's also obviously another regulatory layer that we should look at for sure.
Do you think that because you were talking about like a universal way of activating immune memory, that targeting DNA methyltransferases would be
I think there was one paper where I'm not sure if it was a DNA methyltransferase now, but there was one paper where it was a histone methyltransferase. I don't remember now, where people showcased that at least in certain setups it can work, that you just like overexpress or activate a certain histone. I think it was histone modifier. Activate a histone modifier, and then you kind of get memory to a certain degree. Yeah. I think the other thing I also want to emphasize here, I think maybe I didn't make that point 100% clear, that this memory can be very beneficial. Like in our case, we all agree it was a good thing to have, but it could also be that it's not always wanted that you get like an increased secondary response. So for example, if the second disease would be an allergy, for example, it might well be that in some contexts, then it's not great to have like an overreacting immune system. So I think there's a balance, and I think we also don't really know yet what what is the good and the bad of innate immune memory, because people obviously they like to show stories where it's like beneficial, but we actually don't. Like, we need to know more about it to kind of really understand it, to make a case of like, okay, should we now modify or like give agents that generally boost your innate memory or not?
Yeah. Thank you so much.
Thank you.
Wolfgang, go ahead.
Hi, thanks. A great talk. Thank you very much. This is
Thank
Wolfgang
you.
Leitner from the NIH. I had a question about the timing, and you may have mentioned it, and I missed the beginning of your talk. The timing between the primary and the secondary infection and the impact on the efficacy during the secondary infection. Because when you think about influenza, the within a certain period of time after the infection, during recovery, you are actually more susceptible to bacterial infections, and that's because of the macrophage subtype predominating in the lung. So, at what point does the innate imprinting kick in and is beneficial versus the immunosuppressive environment of the primary infection causing higher susceptibility?
That's a very good question, but I don't have the answer to it. Like, I didn't do, like, our time was always 30 days recovery of SARS-2 and then challenge with influenza. This was kind of the earliest we did. And this was really because we tried to really specifically look at memory. So we were kind of more worried about like residual presence of inflammatory responses and virus. So we kind of really wanted to at least wait a month and confirm that the mice behave like uninfected by all measures we took. And then we challenged them with influenza. So I didn't do I think that would be interesting if we go, because we always our approach was, okay, how long can we drag it out? So we did like 60 days, still worked, 100 days, not so well, 200 days didn't work. But I think it could be interesting to actually also look sooner and like maybe look at, I don't know, day 15 when the mice kind of already almost bounce back body weight wise. We know from the literature, and we also know from our data, that the virus is at least cleared. But whether then you kind of get the opposite side, I think would be very interesting.
Yeah, I mean, there are studies out there that describe how long the M2 macrophages would be dominant in the lung. So I don't know what the right time point it is, but it would be interesting to know at what point do you actually switch from an immunosuppressive to now an enhanced environment. I had a second question about the clotronate depletion of macrophages. That's a really cool experiment, and I was trying to poke holes into it, playing the devil's advocate. So the clotronate causes macrophage apoptosis. What is the effect of that apoptotic death in the lung? So in other words, are you now getting an inflammatory environment because you're causing massive inflammation, or the other way around, if it is I don't know what type of cell death it causes. Could you argue that it causes an immunosuppressive cell death, and that's actually the reason that you're not seeing the trained immunity effect anymore? So is there some kind of caveat that you'd have to consider? And also, how long does that depletion last? So would you be able to wait longer to allow complete clearance of the dead macrophages before they're being replaced?
I think that's a very good point. I think macrophages, if you do intranasal treatment, they will come back after like 3 to 5 days. So there's like a somewhat a
very
short window. We did not look at the cytokines following clotronate. I'm not sure if people have done this for intranasal administration, because people usually do like IP, like general depletion, which we didn't want to do in our case. I know this doesn't really directly answer the question, but if we just treat mice with clotronate and let them recover and then challenge with influenza, there's also no memory. So there's not like some cytokines induced that influence the memory per se.
Oh, perfect. That's the answer, I guess. That's
the other thing that we did was we also depleted T cells independent of clotronate to kind of assess because there's more tissue resident T cells in those recovered animals. So first we thought that probably the T cells contribute, or there could be cross-reactivity, who knows? But it doesn't matter if you deplete T cells or not, for example. So I think we really, like with the depletion experiment, the transfer experiment of macrophages and the T cell depletion, I think we're pretty sure that the macrophages are at least mounting the majority of this antigen-independent memory.
Great. Thank you. And also, by the way, a follow-up to the one of the previous questions, we do have ongoing programs to find out which of the vaccine adjuvants that are out there can actually mimic that effect, so that you don't need a live attenuated vaccine to replicate this effect. Thank you.
That's exciting. Thank you.
All right. I think there was a question in the chat a while ago. I can read it, or if Christian wants to ask it live, let me know. Okay, I'll just read it. So, oh yeah, here he is. Okay, cool.
All right, nice talk. To some extent, Wolfgang already answered that. So I was just interested because sure you have your ongoing infection and boosting in the human population. That's why I was just interested. But I think you were part already answered that there's sure you have some effect, but how the molecular effect actually translates to the immunological outcome is of course another, which I guess is a follow-up.
Yeah, I agree. Yeah. Thank you.
All right. Thank you.
Cool. Thank you, Christina.
Hi, great talk.
Thank you.
Really cool to connect the genetics with the immunology. I was wondering, we are always exposed to viruses. So do you think that there are different types of viruses that cross-protect more than others, or does it have to be kind of a completely new infection that resulted in such a response, and like the ones we kind of know already, they don't protect us from new ones anymore?
I think, it's just like speculating now. I would think that it doesn't matter what the pathogen is, but it depends like which disease, like what the disease properties are of the pathogen. Like two viruses that replicate to similar levels, like they produce similar levels of PAMPs and cytokines, I think they would be the same potent in terms of like inducing innate immune memory. But there's certainly like people have described like, or also I use like different kinds of viruses in mice and kind of tried different combinations, which I didn't show today. And for example, influenza is very good at inducing memory. SARS seems to be very good at inducing memory, but other mouse viruses, such as, for example, LCMV, I don't think they're very good at inducing memory. And I personally don't know why that is. But I think that would be one angle also to find out like what kind of are the molecular drivers of memory. If you have like a scenario where, it's also an infection that is efficient, but it doesn't lead to memory. So what is the difference? Why does one lead to memory and one not? And one, for example, I brought up the LCM example and also mentioned it before. That was a part of the reason why we thought maybe tissue damage is a factor, because like there's some viruses that cause much more cell death than others. And maybe that's actually in mice or in vivo, one of the drivers of this innate memory. But I think we don't yet know fully.
And do you think this process can play a role in long COVID? Would that be something to look into, like to figure out what happens there?
It could be, yeah. I think again, also goes a bit towards my previous point, like that this innate memory or like epigenetic imprinting is not necessarily always good, because it could also mean like what you said, that like some cells are like overreacting when you don't want them to overreact, or you don't want them to react at all. So I think we definitely need to understand much more. I could well imagine that like there's a component also in long COVID where this plays into in a certain amount of unlucky patients, unfortunately.
Thank you.
Another question from Lucas. Go ahead.
Yeah, if I'm allowed to ask one last question. I didn't want to play devil's advocate first, with asking about TRM cells in the lung.
Yeah.
But now, as you mentioned, I was wondering how did you get rid of them? Did you deplete it via intranasal administration of anti-CD8 or anti-CD3 antibodies?
We actually did IP CD4, CD8 antibodies, but we also like did the controls that this also, because I was worried that it doesn't target the TRMs in the lung, but it actually is pretty efficient in also depleting the TRMs.
For a short time point, or because you also get rid of everything that's coming from the lung draining lymph node, right?
So what the depletion experiment that we did was we let the mice recover for 30 days, then we depleted with like 2 doses. Then we confirmed that they're basically gone. And then we waited like another 30 days for like the other T cells in the mouse to come back, because we didn't want to have like basically a functional T cell knockout. And then, so we waited 30 days for the T cells to come back, and then we challenged mice. The idea was that we basically depleted all previous memory cells. And then we still saw that the mice displayed this memory effect. So then we concluded that it seems very unlikely that the T cells actually, memory T cells contribute. Doesn't rule out like a bystander response of naive T cells, for example, but it rules out the memory bystander response, I would say.
Oh, that's interesting, because I thought in the lung tissue it would be hard, to get rid of them, but it seems it works.
I also thought so, but it worked quite well in mine. Maybe I used an excessive dose.
No, but there is actually some literature that lung TRMs do migrate to the lung draining lymph node, so they are not forever. At least for CD8.
I mean, there is a paper where it like the parabiosis, where actually I think the lung, they are most mobile, I think, if I remember correctly, between circulation. Yeah,
exactly.
So maybe that's why.
But there is no way to do this with a rag knockout mouse, and
no, because the rag knockout mouse the problem is that if they wouldn't survive the first infection, basically.
Yeah, maybe with the pathogen that's not so lethal, right? I was thinking about that, like maybe LCMV, I don't know. But as you mentioned, it doesn't cause the tissue damage that's most likely necessary for inducing the memory, right? That doesn't work. But I was thinking something that's not that lethal on the rat knockout mouse, that would be also an elegant approach.
Yes, I agree.
Yeah. Sorry for taking so much time. Thank you.
No, thank you.
The more question, the better, I'd say.
Yeah.
So I don't see any other questions at the moment, but I had one question I was curious about. So maybe a little bit from like my computer science, big data background. Do you think we could eventually get to a point where maybe you can build a map of how similar different pathogens are with respect to the characteristics or what kind of immune response they cause? and maybe use that as a way to essentially cover a broader range of vaccines or other ways to protect from these diseases. What I'm thinking of, I don't know, maybe you have a virus, not very common, you want to go protect against it, and you kind of have a map that tells you which other pathogens are closest to it. But could you, in theory, just use these three vaccines to also protect against that other pathogen where maybe we don't have a vaccine, or it's not important enough to develop a new vaccine?
Yeah, I think maybe both is true. One, that if you just know, like, if the pathogens are similar, if they cause similar disease, I think that's more risky. Like, I think you need to make more assumptions for that. But if you, for example, just have an individual that is infected, if you just compare the disease phenotype, that I think would tell you more. If like a disease phenotype is similar for a new virus to an old virus, then I think you can maybe infer like what would be an efficient therapeutic. I think the disease phenotype is more important than the actual pathogen in that case. But maybe to your point, I think there's like also with these data I showed with this where we sequence every single cell. I think there's like a lot of studies that do this now, and there's like a lot of publicly available data. So I do think it's like a real treasure trove for like people that are computationally savvy, not me, but like there's a lot of data I think that's hidden already, like somewhere, where you could just like, if you could integrate all those different gene expression data from different model systems, different time points, etc., patients, model organisms, I think there could be something where you could indeed like pinpoint, okay, this is like a core transcriptional program or core gene expression program for these memory effects. But I think no one has yet undertaken this task, but I think it will be very, very useful and good to do. Yeah.
Cool. Thank you. Wolfgang, another question from you. Please go ahead.
Since you invited more questions, I'm going to take that opportunity and follow up on what you just said about the this idea of a landscape. So here's the issue. Alexander, you mentioned some pathogens inducing durable immunity, others are really lousy at doing that. And traditionally, we've been thinking of this in the context of immune escape. So you are, as a pathogen, you're not interested in inducing a strong immune response or a protective immune response, because that prevents the host from being it prevents it from being reinfected. So there's a whole bunch of immune escape mechanisms, and we've traditionally been thinking of escape at the T cell level, at the innate immune level. What we have not been discussing in that context is the inability to induce trained immunity as an escape mechanism. So if you look at different pathogens, we have to add that to the list now, where the reason you are getting reinfected or not reinfected is a combination of all of these factors. And it's going to be really hard by comparing different pathogens that trigger the same PRRs to say, well, they're good at inducing trained immunity just based on their PRR patterns, because that leaves out any other adaptive immune escape mechanisms. So it's going to be a combination of all of those. So the landscape, where the catalog would have to take all of those into account. And it's probably much easier to go back to to deconstruct the pathogen and say which ones are the PRRs that are good at inducing trained immunity, and let's use those in isolation. In other words, turn those into adjuvants and therapeutics, rather than thinking about it in the context of the whole pathogen. Because you think about measles. Measles wipes out your T cell memory. Completely different kind of escape than some of the other pathogens. And I was wondering what your thoughts are.
Yeah, I agree with you. But I think one thing I wanted to add was that, at least in our model, so I didn't show any viral loads today, but in our model, this trained immunity does not lead to drastic differences in viral loads, if at all. So it's not really like, if we think about T cell evasions, like the virus needs to evade sterilizing immunity. Whereas here, it's more like
immune tolerance.
One could also say it's more like a tolerogenic or host virus cooperative response. So I think in that case, that would argue a bit against your point of like, do we need to take this into account of like kind of the arms race of virus versus host? Because maybe it's actually even in the virus's favor. You know what I mean? To exist without severe disease, because the host will not die. So it's actually good for the virus in that case.
So it is an escape mechanism, but the other way around.
Yeah, the other way around. Yeah.
Okay.
But again, this combination of pathogens, right? So I don't want to make like a general statement, but I do think that's what I think is an interesting thought of like how to go about this kind of also train immunity and how to prevent disease. I don't want to necessarily say prevent. Yeah, I mean, what you do is prevent disease, but you don't necessarily like eradicate the pathogen. It's more like about a disease-centric way of thinking, I would say.
Okay. Thank you.
Thank you.
All right. Perfect timing. So exactly 1 p.m. Eastern time. That's awesome. Any last remarks, Alex, or anyone else?
I'm good. I'm just very happy to be here, and thank you all for your questions. That was really exciting.
Likewise, super exciting talk, a super interesting discussion afterwards as well. So I think that shows everyone really enjoyed the talk, and quite some exciting, things to look forward in the future as well. So thanks again, Alex, and have a great rest of the day, everyone.
You too. Bye-bye.
Bye.